I'm going to kick off with a question to the full panel. As I said, last year we started with the question, can we do better? Right. So I'd like to kick off our discussion to get the broad lay of the land. And Christian, you're the big face on my screen now, so you're going to be lucky number one. And he has a great smile, ladies and gentlemen. So this is awesome, right? I'd like to kick off our discussion by assuming we absolutely can do better, right, Christian? But my question for you, right, is going to be, when it comes to process development broadly, what is one specific area that you think we absolutely can and must do better in the near future or yesterday, right?
Thanks, Anna, for the question. So I would say scalability is still a big topic with regards to process development and especially downstream processing, where many processes rely on column chromatography as purification method. That definitely is a bottleneck.
Okay.
It's working currently, and process work nicely, but for us, it displays a bottleneck, and especially for therapeutics. And at Athris, we are working on therapeutical applications. I would say a lot can be done better. Yeah. We have been tackling that topic since the beginning because we knew for therapeutics that must change, and we have found solutions for that. I think we'll come to that during our discussion later. But I think that's my favorite for that question.
I love it. All right. So scalability is one of those areas. Is it so much a challenge of achieving a larger scale or smaller scale at higher quality? What do you see as or is it just the fact that we can do a wide range of scales?
Yeah, I think it's a natural limitation which comes in with the column chromatography, where you just are limited in that, and then you start scaling out. Of course, scale up is still possible, but if you go into, let's say, kilogram qualities, that may be a limitation, and we've simply removed that from our processes and still achieve the same quality as we get with other processes. That's if we do that, it's fine, but I think the field will develop in that direction in your future.
Perfect. All right, so we've got scalability. Stu, I want to jump to you and ask you the same question, right? What is one broad area, right, where you think we can be doing better today or need to be doing better?
Okay. Yeah, so definitely agree with Christian for sure. Scalability is kind of one of the big topics as we make more and more products and reach more and more patient populations, larger patient populations. But I guess a second answer would be kind of the analytics and characterization, because analytics and process kind of go hand in hand.
Yeah.
How do you know you're making a better process if you have no measurement of that? So
there is that. Yeah.
A lot of the methods right now, especially for mRNA, like capping and tailing methods, are LCMS based. They're not, exactly fast or easy to develop, or they're more product specific too. They're not very good platform methods.
Okay.
And so I would say, improvement in that area would definitely help with process development. Understanding your product better is always key.
Beautiful. Beautiful. Okay. I think a lot of folks on this call would also agree with that. Yeah. Marc, you are repping the circular RNA world for us today. So I'd love to hear your perspective on this question, but geared towards, right, the circular RNA world. So where is an area that you think, as it relates to process development, we really must and can and should be doing better today?
So I guess we went alphabetically because the two answers before were very good and a lot easier and were on my list.
You're allowed to agree with them. That can be your answer.
Well, I definitely agree with them. Circular RNA, I mean, there's one big difference. We're making circles as opposed to linear. But I think from a high level perspective on improvements, we don't differ all that much. It's more specific, I think, in the IVT, the generation of the actual construct. So I think maybe to go in a little bit different direction than the ER or the mRNA and the analytics. I think an area where the field really needs to develop is in the final formulation, and trying to get a lyophilized formulation for LNPs. I think that'll have a huge impact on transport, cost, storage. So I think that's a big opportunity.